for the invitation and having me here. So as you said, I'm Jessica Garzke. I am a postdoctoral research fellow at the University of British Columbia, and I'm collaborating with the Hakai Institute. And today I'm speaking to you from where I live on the traditional, ancestral, and unceded territory of the Musqueam, Squamish, and Tsleil-Waututh people. And I also did my field studies I want to talk about today in their territory. Today, my title is Running the Trophic Gauntlet, Regional Oceanographic Variability Drives Condition of Juvenile Sockeye Salmon. I'll just give you a short overview where we are all located. You're down here at Dufon Island Sea Lab in Alabama, and I'm way up north in Vancouver, and my research area was up here in the north. And just give you a short introduction about the importance of salmon, especially in British Columbia, the First Nations have a deep spiritual relationship with salmon. The First Nation communities are founded on traditional fishing grounds, and many treaties revolve on the fishing quotas for British Columbian First Nation communities. But also salmon is very important for the ecosystem. They play an important role in the food web, especially for top predators like bears, and also the killer whales. And they are also an important factor. They play an important role in the recreational salmon fishing. And the impact of recreational fishing is about 713 million per year in British Columbia. And just compare it to Oregon, one of the few states in the US that also do have recreational salmon fishery. They have an output of around $285 million US dollars. But also salmon is important for the commercial fisheries. And also here, the annual output is around 324 million. And in the rest of Canada, it's only $180 million. In the past years, we had disastrous declines in sockeye salmon return numbers. And over the last years, there were so many headlines about the low numbers of returning sockeye salmon. And because they play such an important role, we started to think about, okay, what is actually going on? And so what we did is, what did I do? I concentrated on the Fraser River sockeye salmon, and as you can see here, is one of the largest rivers in British Columbia, going all the way from the mid of central BC all the way down to Vancouver until its mouth into the Strait of Georgia. The Fraser River sockeye salmon consists of 24 stocks from 22 lakes and 2 rivers. And just to have a short overview into the decline in numbers, I have here a timeline of the return rates and the abundance of Fraser River sockeye. And you can see that it was really fluctuating over the years from the 1900s and very low numbers in the 1920s. And then it increased slightly in the 1980s. But especially in 2007 and 2009, only 1.5 million sockeye salmon were counted. And this was not enough, but also in 2015, 16, 17, and also 2019, the numbers drastically kept at a very low number. And the lowest number was reached in 2019 with just 620,000 fish of the Fraser River sockeye salmon. So in 2012, when all these low numbers reoccurred over the last years, there was a commission called together. And this commission, it's called Cohen Commission, tried to get recommendations out to researchers and try to figure out what is actually going on. So one recommendation was to investigate where and when significant mortality is occurring during the early marine migration along the coast in British Columbia. And especially it was figured out that the early marine phase in juvenile salmon is one of the most vulnerable and the highest mortalities actually occur. But why? It was not really clear. So just give you an overview about like the general life cycle of salmon. Depending on the species, the fish spend up to 3 years in the rivers and then in the freshwater phase until they actually migrate to salt water, where they spend 1 to 5 years in the open ocean until they return to reproduce again in their natal river. So where I did the study, you can see here on this map the Fraser River outflow, and then the different, the Strait of Georgia here, the Pacific Ocean and Vancouver Island. And especially the Fraser River sockeye salmon, they have the opportunity to take 2 routes to get to the Pacific Ocean. One is going south through the Southern Salish Sea and Puget Sound out to the Pacific Ocean, but also going north all the way through the Strait of Georgia, through Discovery Islands, Johnstone Strait, Queen Charlotte Strait, and then to the Pacific Ocean. And interestingly, like 80% of the Fraser River sockeye salmon, they are taking the way north and not the shorter one south. But also when you zoom in into the Strait of Georgia and the northern and Johnstone Strait, there are different ways the sockeye can actually migrate to get to the ocean. So here's now the activity coming in. I want to ask you, what is your opinion why the salmon actually decreased over the last years? Like, what can you think of? Why are there this high mortalities? Just give me a word, submit it there, and I hope it will work. We will see that you use this and we get a very nice word cloud. And I'm not sure. I tried it just once. Like, just any idea what actually can happen without knowing the actual biological oceanography and physical oceanography of this area. But I think there might be some nice ideas coming up. Sure. Now everything is just, wait, my computer is declining to work. Okay, interesting. Show these boxes. Okay. Okay, does not work. Okay, that's sad. But anyway, just get further into it. So one idea was what was hypothesized in 2014 was the Trophic Gauntlet hypothesis by McKinnell. Pollution, by McKinnell in 2014. And this is just based on the physical oceanography in this area. For one, the Strait of Georgia is defined as a high productive area with lots of nutrients, a lot of chlorophyll A, and a little bit warmer temperatures. But the Johnstone Strait was defined as a low feeding opportunity area because it's a high turbid environment. A lot of vertical mixing is happening there, low nutrients, low chlorophyll A concentrations, low zooplankton biomass available in this area. And then the Queen Charlotte Strait was defined as a kind of a recovery area because of high nutrient availability. Again, high chlorophyll A, a lot of zooplankton, and slightly warmer temperatures. So the Johnstone Strait was defined as this trophic gauntlet or bottleneck, ecological bottleneck for migrating juvenile salmon. And we looked, and researchers looked at the gut fullness of juvenile salmon. But in 2013, Price et al. Looked at the gut fullness in the northern Strait of Georgia, and Brodeur et al. In 2007 in northern British Columbia. But everything what was in between, what was actually happening in the Trophic Gauntlet, was not known. So a master student of my lab looked at the gut fullness of the juvenile salmon while on this migration north to the Pacific Ocean in juvenile salmon. And what she found is that there are actually full stomachs in the northern Strait of Georgia and also in the Queen Charlotte Strait in northern and very north tip of Johnstone Strait, but in between the stomachs of juvenile salmon are very empty. There's like so empty that they're the idea was, okay, they are starving. There's actually nothing to feed on in Johnstone Strait in this trophic gauntlet. So what I did, I did a field study in 2015 and 2016 in the northern Strait of Georgia, Johnstone Strait, and Queen Charlotte Strait. And first, I just used the length and weight measurements and plotted the length and weight residuals, which means it's everything what is greater than zero, means the fish are actually heavier than expected based on their length, and everything what is below zero means they're skinnier or like not heavy enough based on their length. So in 2015, the blue dots seemed they came in a very good shape into the northern Strait of Georgia, lost lots of weight in Johnstone Strait, and then also when they came into Queen Charlotte Strait, they were still lighter, but not too light based on their length. But in 2016, they were in such a bad shape, they already arrived in the northern Strait of Georgia, just very skinny with not a lot of meat or fat stored in their bodies. What I also measured is the RNA to DNA ratio. What this measurement actually means is, or the idea behind this ratio of RNA to DNA is that the DNA concentration in a cell stays constant, whereas the RNA concentration is fluctuating based on the metabolic activity of the organism. So if we have a lot of RNA in a cell, it means this cell is metabolic active, means it's in a good condition, the organism is growing. Whereas when it's really low, the organism is not metabolic active, is not able to grow in a very good shape, in a very bad shape, sorry. So when I did this analysis for 2015-16 along this migration route, I found the same pattern, which follows the gut fullness data of Sam, of my master student. And we found that they came in 2015, the sockeye salmon arrived in a very good shape. They were not metabolic active and not growing Johnstone Strait, and were able to recover and go back to normal, even better, into a better shape in Queen Charlotte Strait. But in 2016, this was not the case. They stayed in a very good shape, could recover a little bit in Queen Charlotte Strait, but still like way lower than compared to 2015. So then I asked myself, okay, what is actually causing this low condition in the fish? Could be food biomass, as we expected from the gut fullness data? Is it maybe also difference in prey species composition in this area, or is it the food quality? So we created or started this juvenile salmon program, which is still running since we started 2015 with sampling the fish in these 3 regions. So we go out with a boat, we use a purse seine to collect the fish that we spotted on the surface of the water. Then we collect the juvenile salmon. but also other fish during these years, why they are migrating between May and July. So depending on what's going on, with the food biomass, I measured the zooplankton biomass, but I concentrated only on the prey species that we found in the guts of the juvenile salmon. So because there are also so many zooplankton species in this area which are not really important as a diet or as prey for the juvenile salmon, because they're vertically migrating. So what I did is I plotted the zooplankton biomass for both years. And what I actually found is that there's not so much zooplankton biomass in the northern Strait of Georgia for both years, but this changes and is year dependent in the area. In Johnstone Strait, it's still the zooplankton biomass really low, comparable low to to the northern Strait of Georgia, but then we have a high zooplankton biomass in 2016 in Queen Charlotte Strait, and still very low zooplankton biomass in 2015. And this is very interesting, because it's zooplankton biomass, it should be the other way around based on what we saw in the stomachs. So I also looked at the zooplankton abundance, and we see a completely different picture. So very low numbers still in the northern Strait of Georgia for both years, but high in 2016, and very high numbers in zooplankton in Johnstone Strait. And then it drops down to Queen Charlotte Strait. And then I asked myself, okay, this does not really match up, abundance and biomass. So what I did, I estimated the average zooplankton size based on the abundance, like the number of individuals and their biomass. So this average estimated zooplankton size indicates that we have larger zooplankton individuals in the northern Strait of Georgia and Queen Charlotte Strait, and smaller individuals or zooplankton species in Johnstone Strait, and that there's not a big difference between the 2 years we measured. And when we think and compare and put everything together, our data, what we saw in the length weight residuals and the RNA DNA ratio, it still does not match up what we see in the difference between years. Because 2015, the fish were in a way better condition compared to 2016, but we don't see this just based on the prey and the prey size and the biomass. So this kept us thinking, okay, we saw this change in the zooplankton size, but maybe it's just different species composition in these areas. So what Sam did in her master thesis is she looked in the prey species composition in the stomachs between northern Strait of Georgia and Johnstone Strait. And what she found is like a very comparable picture for the Johnstone Strait here. It's like very small species, mostly collonoid copepods in the stomachs. And in the northern Strait of Georgia, it's like very diverse between 2015 and 2016. And there are larger species like decapods. We have euphorsids, which is yellow, and larvaceans, and also a lot of cladocians, which changed a little bit over the migration period, but still pretty diverse. So, okay, we see this change in the prey species composition, but there's not this big difference between both years. So still, there's this question, okay, what is actually going on? Why do we see this difference and the better shaped fish in 2015 and 16? So what I did, I just looked at the overall prey species composition, what is available in the water column. And I found again, okay, Johnstone Strait 2015-16, there's really not much going on or a lot zooplankton available that is of interest for the juvenile salmon. In 2015, Queen Charlotte Strait, there is a lot of zooplankton available that juvenile salmon are interested in. And in the northern Strait of Georgia in 2016, this was also the case. So it's a really diverse picture that 2015, some areas it's better or worse. So really a lot going on. So we thought, okay, it's not really the species composition that seems to be very important here. It's not the zooplankton biomass. So it just looks at the food quality. So, and with food quality, I just mean fatty acids. And just to give a short introduction to fatty acids, we have this carbon. These are like a lot of carbon hydrogen atoms and the COOH tail at the end. And when this actually just with in the single bounds is called a saturated fatty acids. And when we have a double bound in here, and it's like this edgy curved form, then this is an unsaturated fatty acids. And depending on the number of double bonds, we have monounsaturated fatty acids or polyunsaturated fatty acids. And part of these polyunsaturated fatty acids are also the essential fatty acids, which means that these fatty acids cannot be produced by consumers like humans or fish. They are actually produced at the base of the food web in the phytoplankton and then transported up through the food web by trophic interactions. And also why fatty acids are important, because energy can be mobilized from these fatty acids in the form of ATP. So the first component, which is really important and easy to access, are the saturated fatty acids. There are no double bonds, so there's not much energy needed to actually form ATP from saturated fatty acids. The next easiest form are monounsaturated fatty acids, and then in general, the polyunsaturated fatty acids. So using this information about the fatty acids, I generally plotted the total fatty acid profiles of both years and the three regions and wanted to see, okay, are there like differences between both years? But as you can see for 2015 and 2016, the picture is not really different for the regions. Yes, the northern Strait of Georgia is a little bit outside, and Johnstone Strait and Queen Charlotte Strait are more similar, but still like not significant between both years. So this is why I looked into the single profiles and groups of the fatty acids. And just to give you an example of the amount of all fatty acids or the total fatty acid concentrations, I show you this. On the x-axis, you see again the different regions. And as before, blue is 2015 and yellow triangles are 2016. So in 2015, the fish, so these are fatty acid profiles from the fish, from white muscle tissue. So what we saw is in 2015, all the fish we measured had a way higher concentration in fatty acids compared to 2016. So they accumulated way more fatty acids on the way north, and then were able to mobilize, or they had to mobilize energy through going through Johnstone Strait, this bottleneck region, and then going to Queen Charlotte Strait. Whereas in 2016, it got a little bit higher in this bottleneck area, but really stayed low really for 2016. And this is the same picture we saw in all like bigger groups in the fatty acids. We see the same picture for the saturated fatty acids. They came with way more saturated fatty acids into the Strait of up to the northern Strait of Georgia. This is also true for the monounsaturated fatty acids and polyunsaturated fatty acids. So 2015, the fish were able to accumulate this form of energy. And this actually fits the picture, or could be a good explanation why the juvenile fish were in a better shape in the northern Strait of Georgia in 2015. Just here as a reminder, the RNA-DNA ratio, and that they were losing their condition in Johnstone Strait because was not enough food. They were starving. They had to mobilize this energy from saturated fatty acid, and then were able to recover later in Queen Charlotte Strait. We see not the same recovery in our fatty acids, and this is just because fatty acids response time until you see a difference is like between 1 and 2 weeks, where RNA DNA ratio response time is between 3 and 5 days. So there's a slight difference. This is why you see this difference in the between fatty acids and RNA DNA. So again, this might happen because in this area we might have different phytoplankton species compositions, which is then transported to the zooplankton and then to the juvenile fish. So it might be more important what kind of food quality you have in response to condition and how to go through these bottleneck areas. But what is also very important is that a study by Minna Hiltun in 2019, she compared the DHA-EPA ratios of different zooplankton species. And just to give you an introduction why DHA-EPA is so important, both of these fatty acids are essential fatty acids, and they are used to as an indicator of food quality in aquaculture. And it says when you have a high DHA-EPA ratio in the food, this is a good indicator. This is very healthy for the consumer. And so she looked at the DHA-EPA ratio of all these different zooplankton species. And what you can see is that there's a high variation in this ratio of these two essential fatty acids. And I was wondering, okay, how much does it really matter? How does it translate? What if a juvenile salmon feeds, like on what kind of species, and how does it translate to the condition and also to recover from low food quality? So I did some literature research to see, okay, do we have actually a time series where we see changes in the diet composition of juvenile salmon? And Prakeshot et al. In 2010 showed this very nice diet composition data from covering the 1960s up to the early 2000s. And what you can see is that over time, the diet really switched. In the 1960s, a lot of insects were found in the diets of juvenile salmon, and some copepods, the black bubbles, but mostly insects. And later it shifted to more amphipods. These are the open circles, a lot of amphipods, and also euphausids, which are the horizontal bars here. So I wanted to see, okay, how does this change in this diet composition how does this affect the juvenile salmon? So I did this analysis and tried to plan an experiment to test this different DHA-EPA ratios and to manipulate the food quality and test how this affects juvenile salmon. So this is the diets I tested. I used as a control commercial aqua feed, which are used in aquacultures. I used krill as the euphausids, and also Artemia, which seems to be a good representation of the insects. I planned this experiment where we fed these 3 diets over a course of almost 2 months to juvenile salmon. We had a phase 1, which was an acclimation phase to the tanks, and then we were weaning the juvenile fish to the different target diets over 1 week. And then we had phase 3, which is the feeding study. And then later, after this 29 days, we switched the Artemia, this low food quality group, to the high quality, to aqua feed, and see if they are able to recover. So I measured the RNA DNA ratio again and found here's the weaning period, and then we started in phase 3, the 100% experimental diets. And what you can see is that the fish that were fed with the Artemia, this very low DHA-EPA diet, directly lost a lot of condition and were less, or not in a really good shape, less metabolic active, and supposed to be, based on the RNA-DNA ratio, not really growing compared to the aquafeed and the krill fed fish. After we switched them, the Artemia fed fish, back to the high quality food, the commercial feed, they bounced back a little bit and were comparable, slightly comparable to the aquafeed and the krill fed fish. Okay, we see a direct response in RNA-DNA to low quality food. I also calculated the average specific growth rate in the juvenile salmon, and we saw that the Artemia fed fish here had the lowest specific growth rate, like the percent of body weight they increased per day, compared to the aquafeed, but the highest, and this is like very interesting, that the highest specific growth rate was found in krill and not in the aquafeed, because I think this is interesting, because the aquafeed is like a designed food which should be, or like provide the best food for very fast growth and very healthy fish. And when we compared it, also I measured the instant mortality rate, and also here you see that fish fed with a low quality food had the highest mortality rate. Just here, I will not go over all this, but I measured like a lot of fatty acids in the diets here, but also in the fish, and try to compare, okay, what is happening in the diet and what do we see in the fish. And here's just an example for saturated and monounsaturated fatty acids, that what you see in the diet is sometimes not what you actually see in the fish, because of the conversion from fatty acid to energy. So when we look at the monounsaturated fatty acids, we actually see that Artemia seem to have a lot of monounsaturated fatty acids, but in the fish, it's slightly lower. And that actually the highest, the aquafeed, which is comparable to aquafeed, have a higher concentration. So when we just look at DHA and EPA, this very important fatty acids we used also to create or to design this experiment, we saw that Artemia had the lowest concentration or percentage of DHA and also EPA. Interestingly, is that DHA was not very different, only that the krill had the highest DHA in the fish, but also in the diet. But what we see is that fish that, or the EPA DHA was comparable in the weaning period, but constantly declined in fish fed with Artemia. The aquafeed and krill fed fish were quite constant, but we see this strong decline in EPA. And why is this important, especially in EPA? Like EPA is like the last resort for fish to mobilize energy from fatty acid to ATP. DHA is usually not used because it's like too energy inefficient to use DHA for ATP synthesis. So it's only EPA. And what we see here is that EPA seemed to be very important, and that the fish needed to go to the last resort and the last source of fatty acid to create energy. And interestingly is also that we fed the fish as much as they needed. So we gave them little portions and until they stopped feeding on the diets. So on the left, you see the Artemia fed fish and how much we gave them for food. And they have the highest feeding rate or the feed intake compared to aquafeed and krill. But when we switch them back, we see that the aquafeed really gave them a lot of energy and were not feeding that much again. So what this actually shows is that even though that the Artemia fed fish had a way higher feed intake, they were not able to compensate this low quality food and the missing energy. or fatty acids to grow healthy and to stay in shape during this experiment. So in summary, from these two studies is that juvenile salmon show clear response to low quality food with lower conditions, that low food quality cannot be compensated with higher food intake, and also that effects of low food quality can be reversed after 4 weeks by intake of high food quality, so they can bounce back. But there are also different, there are also like environmental factors that can directly affect the prey of juvenile salmon and also other fish and fish larvae. These are temperature changes, especially global warming and marine heat waves. And how do you hope this is? What happened is, no, it's not working. Here is, this is just an animation from NOAA, the sea surface temperature anomaly based, shows 2014 and 16. What you can see is what happens in 2013, a marine heat wave started to form, and then getting to the coast of British Columbia, it's like really warm. I just play this again. That in 2013, you see this hot water mass developing in the Pacific Ocean. Here, very good, you can see it, and then slightly moving to the coast of British Columbia until 2015. And this is called the blob. And in September 2014, this blob, this warm water mass, just hit the coast of Alaska, Canada, and the west coast of the United States. And this marine, this blob, this marine heat wave caused a lot of death in fish, but also other marine mammals, because this whole marine heat wave decreased the amount of phytoplankton, of zooplankton, and also decreased the nutrient availability. But also in 2019, another way stronger marine heat wave was observed again. So how does environmental change or temperature affect this, besides ocean acidification? And one way what global warming or marine heat waves lead to is the species shift, that species migrate north to colder environments, but also that species decline. In this figure, you see like very large species in phytoplankton and usually just phytoplankton. So during my PhD, I did experimental studies and what they actually show. And I mostly worked on copepods, and first I started just with a temperature experiment. You see a cooling scenario, ambient scenario, and a warming scenario. And I measured the abundance of acacia, tonsa, in this experimental setup, and found that with warming, the abundance of this acacia, depending on the developmental stage, was decreasing in the warming scenario. Then I also looked at the length, the body length, the chromosome length of these individuals, and found that with warming, the individuals get smaller, like based on the same developmental stage. As you can see here, for example, in adults, we saw this decline in body size. And what I also did was an experiment where I coupled warming and acidification to see, okay, what is actually going on with zooplankton? How is it affecting in the abundance? And we saw this trend in the present 460 microatmosphere environment. We saw this decline in the abundance with warming. But we also saw that with here, the open symbols, the white symbols represent the acidification scenario, that we have had less zooplankton in the tanks. But when we combine this with warming, that this difference between the two PCO2 scenarios is actually less compared to present conditions. And also we see this in the prosome length when we just look at adults. And in the prosome length, we see K in warmer temperatures, which is red. We have a smaller prosome length here compared to the colder environment, and that this difference gets actually way bigger under acidification and with acidification, that they are getting larger again, but still, like with warming, this difference, they stay a little bit smaller in the warmer environment. I also looked at the fatty acid composition and found that with warming under present PCO2 conditions, that we have a lower ratio of polyunsaturated fatty acid to total fatty acid per individual. So they were in that zooplankton had a lower quality in under warming, but that this shifts actually with acidification because we have more carbon in the system, and this changed the fatty acid concentration in zooplankton. So we have to conclude that there are indirect effects by environmental change on the organism. So there we see these effects on the abundance. We see this decrease on the individual size, but we also see that environmental change can affect the fatty acid composition of diets or of prey items for higher trophic levels. And also, and this means we actually have to include the species composition, which has been shown in this feeding experiment with the juvenile salmon, but also we have to think about that we have changes in the individual body size, which leads to effects on feeding and trophic ecology. So, and especially we have to think more about food quality, especially what I showed, mostly fatty acids. So we have to include food quality into ecosystem and food web models. And because currently, most of the time, zooplankton biomass is just included. And as we have seen in my field studies, that is not really sufficient to relate this sometimes to the condition of juvenile salmon. All this work was done with a lot of people, like what I did during my PhD, but also here at UBC with the Pelagic Ecosystem Lab, the Hakai field crew who helped me sampling, and the funding agency in Canada, Mitex, which I was funded by. Thank you very much for listening, and I'm happy to answer your question when I'm able to stop sharing.
I think you touched on this a little bit in your talk, but the annual variability, do you have a sense of, El Nino years versus La Nina years and things like that?
So so there are time series like annual samplings, kind of like a time series in zooplankton and also phytoplankton. We see like a general shift over the last 10 to 15 years, not really with El Nino and La Nina years, especially within the Strait of Georgia. You see stronger effects on the west coast of Vancouver Island, like directly in the Pacific Ocean, but it takes a while until we see these changes in the Strait of Georgia based on the physical oceanography here. But it tends to be a general shift to smaller species, to more collonoid copepods compared to larger species like Metridia or euphorsids. So smaller copepod species, and the same is also for phytoplankton. But yeah.
Also, do you see any changes with their predators?
What we've just like with the juvenile salmon, it really depends on the species. We see strong decrease in Pacific herring, but this is not really related to the El Nino. But there seems to be a stronger effect with the PDO. But yeah, this is all I can say to that. But more like a long-term trend, which is affecting Pacific herring. And we also see more anchovy here, which is interesting in some years. Okay, there's other fatty acid composition in copepods or salmon most similar to a specific algae group. Yes, so the DHA-EPA ratio is also an indicator to the ratio of diatoms to dinoflagellates. They have diatoms have a higher concentration of, now I have to think about, of DHA, and dinoflagellates have a higher concentration of EPA. So a lot of people use this ratio of DHA to EPA to indicate, okay, it's more diatoms or dinoflagellates. The area here, especially the Strait of Georgia, is a diatom dominant area. So people have used the DHA-EPA ratio to look into the composition of diatoms to dinoflagellates, and also some fatty acids can be used as biomarkers for cyanobacteria, but it's a little bit tricky because also what studies have found, it really depends on the nutrient stoichiometry, like if you have higher concentrations of essential fatty acids or not. It's like a little bit variable, but some species can be related to specific fatty acids. Yeah. Can you trace? Yeah. So as I said, DHA and EPA are used as biomarkers, and also other fatty acids like alpha-linolenic acids and other essential fatty acids can be related to algal groups. We are currently working on that. A friend of mine, a colleague in my lab, she is actually analyzing particulate organic matter samples with fatty acids and try to see if that is possible and how consistent that is.
Well, that was a great talk. We really appreciate you taking the time to to chat with us. And too bad you weren't here in Dauphin Island, but if you are down this way, I think that would be great to meet you, and you can meet everyone else.
Yeah, great turn. This would be great. I've never been to Alabama, so this would be a great opportunity.
Absolutely. It is nice to be able to bring speakers from all over, in this way.
Yeah. Thank you so much for having me and giving me the opportunity.